Good afternoon. Oh, yeah, I don't like this one.
Good afternoon.
Good afternoon. Woo! To the 3MT this wonderful Tuesday afternoon. Appreciate all being here, especially the lovely judges, and most importantly, our wonderful competitors, these brave young souls. Thank you for being here for Zoom and TCS. I'm just going to go over a brief overview of what the thesis competition is, in case you guys may be wondering, especially for the benefit of the audience. First off, my name is David. Hi guys, I work for the Graduate College. Pleasure to be here. Appreciate you guys having me up here and hearing my lovely voice this afternoon. Some stats about our lovely grad college, which you guys are somewhat part of the stats, so thank you for that. Give a bit of a competition overview, which I hope you guys are competing. That you guys know the overview is, especially the judges. They'll be grading you guys. And we do have some more heats coming up, so if you guys do have the time, we do have another one tomorrow morning. But most importantly, the winners of our heat today will be going on to the championship this Friday afternoon from 6 to 8 p.m. Hope you guys can come out and support our fellow graduate students. And we'll be tested 3MT, started in Australia, and then now we're here in South Florida. So amazing travel. Next, we're going to briefly over competition rules. I hope you guys are firmly aware of them. But the most important thing is going to be our judging criteria, which 50% is going to be based on your comprehension and contents, and the other 50% is going to be on your engagement of communication. So be mindful of that and what the scoring will be. So please definitely keep that in mind when you guys are speaking. And of course, we can have a 3MT with our lovely volunteers and sponsors. So thank you everyone for being here and dedicating your time with us. And of course, no 3MT will be without our lovely, beautiful judges. Thank you guys again. You guys are great.
Yeah, the judges.
You guys can see you here. All right, with that being mentioned, we're going to jump right into it. For our first competitor, our first competitor today, Giselle Sim. She is representing the Charles E. Smith College of Science.
Medicine.
Yes, College of Medicine, thank you. Her title will be Effects of Cell Starvation on Neuroinflammation in Alzheimer's Patients.
So according to the NIH, there are more than 6 million people who are over the age of 65 that are suffering with Alzheimer's disease. And that doesn't even account for the ones who are under the age of 65. So I know you probably heard the terms Alzheimer's and dementia just mentioned kind of loosely, but what do they actually mean? Dementia is the overarching umbrella term that encompasses cognitive impairment, while Alzheimer's is the specific neurological disease. So you're looking at specific disease, so we're talking about Alzheimer's. And some of the characteristics of Alzheimer's include memory loss, cognitive impairment, and the inability to perform basic normal human functions such as remembering to eat. And unfortunately, there is no cure for this devastating disorder. But we can break up Alzheimer's into 3 different parts: the early, middle, and late stages. So my research focuses on the early stages of Alzheimer's because it's much easier to treat something in the early stages rather than the late stages, and it's virtually impossible to treat Alzheimer's in a later stage. So with all that background being said, the current findings state that there's a specific peptide called amyloid beta that causes cell death, and that's what ultimately produces or gets someone have Alzheimer's disease. However, when you get older, then there is an accumulation of amyloid beta that just gets presented or just accumulates in your brain as we age. So there is that missing link in between having Alzheimer's and having amyloid beta. So what my lab was doing, we were gonna go back in time a little bit further than when you find amyloid beta, because we're saying that there's a cell starvation hypothesis. So you might be asking, what is this hypothesis? That's essentially where the cells have a lack of nutrients. Specifically, we were looking at glucose. So when you have a decrease of glucose, then it causes a decrease in the glycolysis pathway in microglia cells, and then that was gonna help and that was gonna produce neuroinflammation or brain inflammation in the brain. So, and what we were trying to do, we were trying to model this amyloid beta blocking the glucose. So in order to block the glucose, we used two drug models in order to show that, and then the drug models increased the ATP that was present, and we also found the pro-inflammatory cytokines, which increased in the microglia cells, and that allowed us to show that we can look at the glucose transporter for further therapy treatments, and also at the increase of the intracellular glucose.
Thank you. May we please have a moment of silence for the judges to mark their ballots.
We're having an issue with this iPad. Yeah. We got it. Technical difficulty. Sorry, but
Progress.
correct. Yeah. Perfect. Just for suspense. Like we
Our
knew.
first competitor.
Yeah, we're ready. We're all ready.
What is paper? Yes. Thank you, our judges, for your lovely time. May I please have our second competitor on stage. Our next competitor, Alicia Myers, who will be representing the Charles E. Smith College of Medicine for topic, reducing costs in atopic dermatitis.
Dermatitis.
Dermatitis. Thank you.
Good afternoon. My name is Alicia Myers, and I'm a third-year medical student at the FAU College of Medicine. Today, I'm excited to be talking to you all about atopic dermatitis, more commonly known as eczema. So there's three things I want you to know about this disease. One, it's incredibly itchy. Mosquito bites all over your body type of itchy. Number two, it's chronic. A lot of cases from childhood progress into adulthood. And number three, it's incredibly common. In fact, 17% of children in the United States have this disease, and 6% of people have it into adulthood. So let me ask you a question. How much money would you spend to get rid of a really itchy rash? A lot, right? Well, Americans are spending $5.2 billion annually to try and reduce this disease. And $128 million alone is spent simply on lost productivity with people missing work as a result of this disease. So after seeing this time and time again in my pediatric preceptor's office, I decided to do some more research, and I spent several years in my early medical school education researching how we can reduce costs and decrease the burden of this disease. So there's 4 major ways we can reduce costs. Number one, prophylactic therapy. So studies have shown that by moisturizing our babies, we can avoid them getting atopic dermatitis whatsoever. So this is especially cost effective in patients who are at high risk of developing the disease. So moisturize our babies, number one. Number two, in mild cases of this disease, we need to use corticosteroids. They're a cheap, effective medication to treat the disease and are incredibly effective according to the studies. Finally, thirdly, in moderate to severe cases of the disease, we need to use more aggressive measures that are more expensive early on. By not treating early on to the disease with the more expensive measures, we actually spend more money using the cheaper medications over the longitudinal period. And number 4, education, education, education. As we're all sitting here learning more, we need to educate our patients. So often, patients leave the physician's office not knowing what to do. By educating our patients, we have better results, better outcomes, and spend a lot less money. So, 4 things. Prophylactically moisturize our babies, use low-cost, effective mechanisms of action which can treat the disease early. Number 3, use more expensive disease when necessary, and education. I hope I give you the itch to reduce costs.
Nice. Great job. You handled that so well.
Yes. Please have a moment of silence for our judges to mark their ballots.
So you were heard perfectly without a mic. So if that happens to anybody else, you just ditch the mic. But try to use it if it's working properly, because it will allow everyone to hear you properly.
I got a pretty loud voice. Can I use the stand and then just talk over it?
Yeah, absolutely.
But you want to hold it though, right?
No, not anymore. If it's gonna do that.
So yeah, we'll make sure
my
everyone,
mind's gonna go here.
if anyone wants to stand, we'll make sure that it's set up for you properly. We don't want you to have to figure it out. We'll definitely get it set up. So it's the front part. the front part. That one, in your left hand. There it is. And then you can raise it.
I'm in college, guys. What, you're like.
We'll make sure that it's set up because I want to make sure that for all photo and video, it's not in your way. Yeah, don't be silly. Just hold it. Yeah.
All right, guys, there's the next competitor up on stage.
I'll do the stand.
The next competitor, Emmanuel McKinley. He will be representing the Charles E. Schmidt College of Medicine. This topic: what does a surgeon look like? How stereotypes may negatively impact recruitment into the least diverse specialty in medicine.
Hello, everyone. My name is Emmanuel McNeely. Marion Edelman coined the phrase, "You can't be what you don't see." As a Black underrepresented minority in medicine, I've had a winding road to medical school. What is an underrepresented minority in medicine, you might ask? It's one whose racial and ethnic population in medicine is considerably less than the US population. So I committed to research, and I coupled my passions for community outreach and surgery, and I asked the question, why are there so few underrepresented minorities in medicine, particularly African Americans, pursuing the least diverse specialty in medicine, namely orthopedic surgery? Our results were eye-opening, yet not shocking. So we interviewed 30 undergraduate students from a top-ranked institution. We asked them their perceptions, their opinions, and thoughts towards orthopedic surgery. These students gave us a lot of feedback, and that's what I'm going to share with you today. Nearly half of the students had never encountered an African American or Black physician in their life. Over half of the students had never encountered a Hispanic or Latinx physician, while nearly 90% of all respondents had encountered a white or Caucasian physician. Interestingly, all underrepresented students in medicine did consider a career in medicine in our study. However, only one considered a career in orthopedic surgery. So the big question is, why is this? We asked our students if you could only pick one gender and one racial or ethnic background, how would you perceive an orthopedic surgeon? Almost all of our students said white. Nearly almost all the students said white male, and only one respondent in our entire study perceived an orthopedic surgeon to be a Black individual. Why is this important? As we increase diversity, numerous studies have shown that you have better outcomes for your patients, and you also help eliminate health disparities. Therefore, in my career, I'm committed to continuing this race of increasing diversity within medicine, namely the least diverse specialty in all of medicine, and also improving diversity from pre-med students, medical school, residency, and in academic leadership. Why is it important? I want to make sure that I improve outcomes for all patients in every community, and this is how we do it. Thank you very much for listening.
Thank you. We just have a 1 moment silence for our judges to mark their ballots. Thank you, judges. May I please have our next competitor come to the stage? Our next competitor, Alexandra Dinovic. She'll be representing the Charles E. Smith College of Science. Her topic, nose to brain delivery of therapeutic peptides.
Hi, hello. So what are peptides? Maybe you have heard of amino acids or peptides that as dietary supplements, or peptide in protein shakes, or peptide in face creams. So see, we can use amino acid building blocks to synthesize and create therapeutic peptides or therapeutic proteins as a drugs that we can synthesize in the lab. So therapeutic peptides are an emerging class of drugs that are used more and more in clinics. They can be very effective. They have less side effects and have very little side effects or no side effects. Diseases that affect brain are a major health issue in the world. Why is that? That's due to challenging drug delivery to the brain. The brain has that blood-brain barrier that's kind of like a phospholipid protective layer that prevents any harmful chemicals like bacteria, viruses, any harmful chemicals to reach the brain. So because of that, you cannot deliver drugs to the brain if you take as a pill, like orally, or as injection. However, you can take it as intranasally. So what is the issue? See, if you take a drug intranasally, it is shown that just 1% of drugs gets actually absorbed into the brain. So to improve that nose to brain delivery, my research is focused on new system that is based on this odronolactin, or OL. This odronolactin is cyclic peptide that is actually extracted from the skin of the frog. So see, what is important for that is it's a small lectin and it binds to sugars. Why is that important? Those sugars are located on the nose. So the idea is to synthesize therapeutic peptides with odronolactin, and with that, they would bind to sugars that are located in the nose, increase residence time of the drug in the nose, and the drug would get absorbed into the brain. So my research focused on synthesizing and screening libraries of these therapeutic peptides so they can get delivered to the brain to achieve desired activity. What is also important to mention is that odronolactin is not biologically active in the brain. So we can actually do this. Thank you.
Please have a moment of silence for our judges to mark their ballots. Thank you, judges. May we please have our next competitor come to the stage. Next competitor, Amish Mishra. He'll be representing the Charles E. Schmidt College of Science. Title, So, You Want to Build a Protein?
Imagine with me, you wake up on Sunday morning, ready to satiate your hunger. You heat up a pan and crack an egg right into that pool of creamy, bubbling butter. Breakfast is served with a beautiful yolk and a perfect egg white. But did you ever wonder what makes an egg white turn white? Today, I'm going to show you how the answer to that question is going to open an investigation into the very atoms of our universe. Egg white has a protein called ovoalbumin. You can think of it like a string of atoms that's wrapped and folded around itself in a very precise and complex way. When you heat up the egg, that protein structure falls apart, and the proteins link together to form a net and give the egg that all familiar white color. Now, I use this just as an example to illustrate a very profound point. The structure of a protein determines the sort of properties that protein exhibits. In the case of eggs, the structure determines the color. And proteins are pretty much in everything. They're antibodies serving on the front lines to defend your body. They're enzymes in your saliva and stomach helping you digest food. And now scientists are able to design their own sequence of atoms to manufacture proteins that have never been seen before. But there's also a problem. Protein stability. You see, when proteins fall apart and they can't go back to their original folded state, we call them unstable. And just think about the proteins in the egg white. When you heat up the proteins and they fall apart, they can't go back to the way they used to be. So if we're going to design a protein, we need to know what sort of structural properties the protein needs to have to make it useful in the place we want to place it in. This is where my research comes in. My thesis treats the atoms in the protein like data points and takes a data-driven approach to study the role of structure in protein stability. I've obtained thousands of proteins from scientists in the University of Washington where they've built and studied these proteins in the lab. I've been able to use a powerful tool of mathematics called topological data analysis to analyze the space and the gaps between the atoms. Our preliminary results show that smaller holes and spaces between the atoms correspond with more stable proteins. Knowledge like this in hand with AI is going to be able to aid us in the identification and the creation of more stable proteins. This means that if you're able to make a protein on a computer and simulate it, we can determine if that protein has potential to be useful. Future applications for work like this go into tackling diseases that haven't been tackled before and developing drugs like never before. My research just shows a little glimpse into the intricate, complex world that God has spoken into existence. Collecting data and analyzing it is just one way a researcher like me is able to explore this winter wonderland of complexity. So, do you want to build a protein? Come on, let's go and play.
Great job, Amy.
There's a moment of silence for our judges to mark their ballots. Thank you, judges. May I please have our next competitor come to the stage? Next competitor, Morgan Slivian, he'll be representing the Charles E. Smith College of Science. His topic, what's up your gut? Solving mysteries of the avian gut microbe.
It's what's inside that counts. Don't judge a book by its cover. We've all been raised on these little mantras, but I've built my dissertation around them. You see, almost every living thing, us included, is home to millions and millions of bacterial cells living all over in the parts of our body, forming communities called microbiomes. Researchers have found that the gut microbiome, forming a community in our digestive system, relates to digestive health, obviously, but also aspects of our brain, including behavior, and even cognitive ability. So humans have started studying animal microbiomes to understand how to model them for human health. I'm more interested in studying animal microbiomes, the animals sick. Microbiomes in wildlife populations can explain variation in individual survival, behavior, behavior, and even response to environmental change, like urbanization or global climate change. So for my dissertation, I am studying the understudy, bird bacteria. My goal is to understand how a bird's gut microbiome relates to and may actually influence its brain. My first step was to sample the gut microbiome of our captive population of zebra finches to figure out what's up their gut. And then I measured their performance on various cognitive tasks. And I found out that birds that solved these tasks faster had different microbiome characteristics than birds that solved them more slowly. And those slower solvers had a relatively higher pathogen abundance. So cool, but what does that mean? Were my smarter birds smarter because of their microbiome? It turns out stress is actually a big mediator in this gut-brain relationship because it impacts cognitive performance negatively, which we have all felt, but it also can trigger an immune response. So in trouble for the microbiome. So maybe my dummies were just more stressed out on that day. So my next step was to figure out the impact of stress on the health and microbiome of a wild population of northern cardinals. And I found that cardinals with a more diverse gut microbiome, well, they were better off. They were in better body condition, lower baseline stress hormone levels, and even a less dramatic stress response. And they were sexier. More elaborate beak and feather coloration, which is used in their sexual interactions in the species, showing that beauty really is more than skin deep. Because it's tough to measure cognition in a wild population, I am now back in the lab for my final experiment with our captive population of zebra finches, and I am testing the effects of a well-documented and biologically relevant stress treatment, extended social isolation. I'm testing the impacts of that on the gut microbiome, sexual orientation, and also cognitive performance. Now, zebra finches are like us, they're social, which means that we know from our time during COVID and quarantine that extended social isolation is very stressful. So figuring out what is, how stress impacts those, those most important to us, it can really have long-lasting effects both outside and in.
Yeah. Good job, Morgan.
And please have a moment of silence for our judges to mark their ballots. Thank you, judges. May I please have our next competitor come to our bench. Next competitor, Saad bin Saeed Ahmed, will be representing the Charles E. Smith College of Science. His topic, prediction of radiobiological indices for head and neck cancer patients using artificial neural network.
Head and neck cancer is one of the most common cancers worldwide, and its incidence rate is so high that it's reaching up to 800,000 patients annually. So it accounts for around 4 to 5% of total cancer burden in the United States. So more than 50% of the patients have to go through the radiation therapy at some stage of their treatment. And radiation therapy in head and neck cancer is a quite complex and sophisticated process because tumor usually lies in the middle and in peripheries there are too many healthy organs. So it's a quite a challenge for the planner to make an optimized plan in which it has to give high dose to the tumor and preserve or save the healthy organs. So these treatment plans and strategies were evaluated by using the radiobiological indices in the clinics. So these are basically mathematical indices which are basically expressing that how good you are treating the tumor and how good you are preserving the OARs or healthy organs. So in conventionally, these indices were calculated using software or spreadsheets, but in these spreadsheets and softwares, the background data of OARs and tumors were quite old, and they were calculated using some experiments like 30 years ago. But now there's a need of personalized indices which were calculated or which were expressing those patients which were treating now. So in this research, we developed an artificial neural network to predict the personalized radiobiological indices by using the patient demographics, patient tumor specifications, histology, and how tumor is near to an OAR, what is the distribution with the OAR and the PTV. So we account all these information to predict the radiobiological indices. And currently we are doing this only for the parotid glands, but in future we have plans to extend it for the cochlea to avoid the hair impairments. And another thing is for submandibular gland. So we hope that this tool will empower the clinicians to make better treatment plans and make the toxicities less as much as we can. Thank you.
Thank you. Maybe have a moment of silence for our judges to mark their ballots. Thank you, judges. May we please have our next competitor come to the stage. Our next competitor, Sheikh Mohammed Ashrafal, will be representing the College of Engineering and Computer Science. His topic, a healthcare wearable belt for monitoring parameters related to health failure.
Let me start off with a question. Would you think that your car would be as good in performance as it was before having an engine failure? Certainly not, right? Now imagine, what would be the impact if the engine of the human body, heart, fails? Heart failure is a structural abnormality of the heart where heart is not being able to pump enough blood to meet the body's requirement. And if it happens, it can not only decrease the efficiency of the heart itself, but it can affect other organs of the body. Because of this, it makes heart failure as one of the most leading causes of death around the world. Now you must be thinking, and rightly so, there must have been some solutions to this pressing problem. And yes, there are. There are two existing solutions, but unfortunately, both of them are implantable solutions. That means that not only they are expensive, but they are also involved with having risk of surgery. And on top of that, one of the most widely used and standard solutions for the heart failure, known as implantable cardiofib, is only available for critical patients of heart failure. That makes around 50% of 64 million heart failure patients around the world. So there's an urgent need for a solution that is non-invasive and for all heart failure patients. Because in the end, all lives matter, right? So my thesis is based on the development of a non-invasive healthcare wearable belt that all heart failure patients can wear. The wearable belt that we have developed measures and monitors different parameters that are related towards the monitoring of heart failure. These parameters include measuring the resistance of the thorax region of the human body, ECG, heart rate, and detecting the motion activity of the patient. These parameters are shared with the patient's doctor using a mobile application that is Wi-Fi enabled. The application will be accompanied as part of the next step with an artificially intelligent algorithm that can generate alert whenever one of these parameters crosses their say threshold. The algorithm will help in predicting the failure so as to stop the engine of the human body from failing. Thank you.
Please have a moment of silence for our judges to mark their ballots. All right, thank you. For our last competitor, may you please come to the stage. And for our last competitor this wonderful afternoon, Daryl Diajus. He is representing the College of Engineering and Computer Science. His topic, point of care vascular occlusion tester for sickle cell disease.
So approximately 100,000 individuals in the United States of America, as well as millions worldwide, are affected by a genetic disease known as sickle cell disease. So for those of you who aren't familiar with sickle cell disease, what happens is under low oxygen conditions known as hypoxia, rigid fibers begin to form within the red blood cells, and this stretches the cells and gives it the iconic sickle shape that the disease is named after. Now, why this is important is because normally our cells are very fluid. They're able to squeeze and contract between the smallest of capillaries and deliver oxygen to the various parts of our bodies. However, in those low oxygen conditions, those with sickle cell disease, their cells can cause an occlusion within their vessels. So this is what's known as vascular occlusion. So my research is giving those with sickle cell disease the ability to manage their situation. So currently there are no devices on the market similar to those who have diabetes who can check their blood sugar levels with a glucose meter. We have those with sickle cell disease have no current devices such as that. So here I'm utilizing what's known as microfluidics, where we use the minimal amounts of fluids to conduct experiments. So if you look at the images over there on the top right, the microfluidic device itself is no bigger than a quarter. Right, we're using a single drop of blood placed into this device where there are micropillars that will act as an emulated blood vessel. So those we can rapidly test the blood of sickle cell disease patients where the cells will pass through these microgaps if they are healthy, and if they are not, they will occlude in the channel. The channel itself is being measured using an electrical signal, and I'm measuring the electrical impedance across this channel, thus quantifying the occlusion. So using the occlusions, the quantified value as well as the rate of occlusion, we're able to give the patients just a sense of awareness towards their situation. So this can potentially be used in clinics for rapid testing, and later down the line, it can be used as a point of care tool for individuals to use at home.
Good job, Dale.
Thank you. May I please have a moment of silence for our judges to mark their ballots. All right, well, thank you, judges. While we got those ballots ready for you guys, is this favor? A very big thing is our People's Choice Awards. Everyone who's currently here, judges, friends at home, family, you guys competitors, you guys are welcome to do the People's Choice Award. Make sure you guys pick me as your favorite MC. But please,
So
everyone here, if you don't mind, that'd be great. I can't choose. I'll pick all of you guys, so no favoritism from me. But yeah, all
Yeah,
deserve
you
it,
can do
that's
this
for
as
sure.
well.
Just take a moment of your time, do this little survey for us or award, and we'll get back to you the results as soon as we can. In the interim, there's some refreshments outside if you guys want while we wait, and restrooms as well. You guys are the dudes.
So we want to make sure all of you vote. Once all the competitors have
Austin's all
placed their
right. Do
vote,
that.
we're gonna do a photo of all the competitors.
But thank you for being here.
Yes, I recommend all of you vote. Everyone's eligible to vote.
We're all dudes. Yeah.
You are on camera.
Good burger.
Whatever happened to Kel Mitchell?
Oh, they did the Saturday Night Live.
With him?
Yeah, like last year.
Did they do
did
all
a Good
that?
Burger.
Did
Hilarious.
they do like all that as well?
No, just the Good Burger. Hilarious.
So do we have all the competitors here? Or anyone step out?
I think everyone's here.
Do your vote. Do your vote. I just want to make sure we get at least one group photo before we announce the winners. You all did really well. All right, let's take a group photo. Let's do it.
So I believe there are 10 of you, 9 of you? Nine. So obviously someone will be in the center. We're not gonna have the banners in there. We don't wanna move them because they're in a good place for the other competitions. But if you're to this side of this line, you'll kind of face this way. And and this side, no, we don't have to do any sorority squats or anything like that. If we're at that angle, if you're on this side, you're gonna face this perfect. No, like a little bit of a U, so you're perfect. And then you guys be angled in this way, right? Perfect. You guys be angled in this way. Then you come up, we'll make a little bit of a U, so you guys can come up a little bit. Perfect. And make sure that we're not standing in front of everybody, so we're over a little bit. But you got them all in the shot? How's it looking? All right, we'll let you know.
Pretty good. Morgan, if you could just move over a little bit. There, perfect. Thank you. You could look at his. He's the main one. Mine doesn't have any film in it. That's just a joke, guys. Coming out good? Yeah. Wonderful. All right. Thank you.
Teach today? This morning. Yeah. Teach tonight. Tonight. Till when? Till when?
Till 9:20. 6:30 tonight. Let them out.
You need to get some energy.
I need some coffee.
I'm going to
get
get.
home.
I know. I teach this morning, and last night I had a international meeting till 11 p.m.
Oh my goodness.
because of time difference.
Time
So
difference, yeah.
my 8 p.m. is their 8 a.m. I would like to do it earlier, but yeah.
Taking language classes right now with somebody who's also on the other side of the world. So it's like, good morning for her. And it's, late for me. And if I miss the normal time I can't have another time. It's like 11 o'clock our time.
Yeah.
I get it. But
It's every Monday. I'm like, okay, Monday is a long day
Long day.
here, and I have to keep extended till 11 p.m. every Monday. Kidding.
like you needed more reason than not like Mondays.
Yeah, I know. I hate Monday. Yeah, Monday is our department meeting all day from 9 a.m. Yeah, department meeting like a committee of student, committee of faculty assembly, PhD, everything is every Monday.
How do you have energy to go through the rest of your week after that?
I know, it killed me right there.
Hi guys, David here. I just want to say thank you all again for everyone for being here. Let's give one more hand to the competitors. Great stuff, great souls. Good luck on your research on top of it. I guess we'll jump right into it. I'm just gonna go down the list as you guys can see here. So our first or our second runner-up shall be Manuel. I know your last name.
That's a real one, that's looking good. Right?
And for our first runner-up, may I please have Morgan come to the stage. And before we announce our winner, we're going to announce our People's Choice Award to who the audience and friends and family at home have selected. Our People's Choice Award for our heat today will be Anush. We're building protein, guys.
Why is that white? Well, no, I haven't wondered that. But now I know. There's a lot else I'm wondering now. Here's
And for today, the winner of our heat will be going to the championship with all the other two people. Our champion today is Amish. Really extra proteins. Thank you guys. You guys are beautiful and wonderful. I also want to say thank you to our lovely judges today, who's kind of anonymous, but not at the same time.
No, they're not.
Yes.